farmers that have like tools to assess what's going on, I think it's like the best way to learn. You know, if you have time to just do a quick assessment of like, oh, I did this. Ooh, here's what happened. Oh, I'm not doing that next year. You know, I think that's the sort of like having tools that are easy to use, but sometimes to complicate it.
Yeah.
You know, and that's okay. I think that's great.
So great practice, but it kind of concentrates all of the nutrients really in the top 4 to 6 inches, which is also kind of where all the microbes will stay. So they're, you know, in our no-till systems, which we're trying to do a lot of in Vermont, it actually minimizes like the amount of carbon and microbes and roots that go into the deeper kind of like sections of the soil. Well, I think anytime you can have plant diversity, whether it's trees or, you know, a lot of our no-till agricultural fields in Vermont are, you know, there'll be corn.
Right.
And not always continuously, but generally for multiple years in a row. So then, you know, the roots of the corn really do only kind of go so deep. And then when farmers are applying nutrients, it's all on the surface. And so it takes time for that to work its way down, where in a tilled system, they'd be kind of incorporating those nutrients and getting them down deeper into, you know, the soil profile. So when you're adding in plants like trees, you know, that's
still too wet to dry on top.
It's like the right time to get there on the field.
They also, a lot of folks will, make
I feel like we can see the root depth really well. We can see it fairly well in Molly Slice, and also if you want to come take a look at the hole, that's also a good way to take a look at the root depth.
In what direction are the roots going? Are they, you know, pretty much just going straight out? Are they going straight down? These are the kind of things that, you know, you look at whenever you do like an in-field assessment. You obviously don't look at that whenever you take a soil probe, take soil samples, and take your soil sample back to a lab. So there's a lot of things that you can see out here in the field that you can't actually see back in the lab, because whenever somebody does a soil sample, usually you have, this is not marked right here, but whenever you take a soil sample, you go to a specific height within that soil probe to make sure that you capture the same horizons every time. And so often when you do that, I'm just going to do it in the plans here. This is a lot tougher. Oh my God.
Molly knows. She dug our hole.
So, in the words of Heather Darby, I'm very disappointed in this group not rushing forward to look at soil.
Definitely there's some compaction.
I think so, yeah. Definitely. Some compaction, there's a lot of things here. So whenever you take a soil sample with a probe, you have to make sure that you remove any plant material, because that's really going to throw off any nutrient nutrient analysis. And you make sure that you can try and get as much of the plant material out of there as you can, including roots. And so they often will take the soil sample and they'll sieve it, and that will take out a lot of the plant material, but also the rocks that are bigger than whatever the sieve is.
I saw you take the soil sample in a group of grass.
Yep.
Okay, but you don't, when you're taking a soil sample, you want excess bare ground is better?
You want to make sure it's representative, exactly, of whatever field you're taking. If you're if you're taking 20 samples in one field and you do it in a bare spot, you make sure you always do it in a bare spot, and especially if the field looks pretty bare altogether. But the main thing that you're trying to do is, make sure you're taking a very typically found looking amount of area.
So a hay field,
yep.
When I do a test, I try to move the grass out of the way to not get that in the sample. That's the right.
Typically that is very much the practice that you want to follow. But if you have like a really like highly covered area and you're not taking soil samples back to a lab, but you're doing an assessment there in the field where you're going to be counting and measuring the depth of roots and also looking at which directions the roots go, then you would want to make sure that you capture a really good representative plant growth formation there.
The depth. Oh, sorry.
I was just gonna say, bring us back to the roots a little bit, but I want you to ask your question.
Oh, my question's about the roots. So what's the significance of understanding the direction of the roots?
Yeah, that's a great question. So when trying to assess kind of the biological health of the soil, one thing that we're going to be looking at are the plant roots. And so there's kind of two things to look at. One is the depth, and then Charles also mentioned the direction. So I will answer your question, Molly, but I'm going to do it in kind of a roundabout way, just to poke at us and make us think a little bit. So if you want to take a look at what we've got in the shovel and also maybe try and see what you can in the hole, does anyone have any observations about rooting depth? I feel like I heard someone say it before. Do you like see a lot of roots in here?
No. Tons, but you can see them sticking
happy
out
to see that.
of the bottom.
Yeah, so we've got some, and they must be going decently far down because looking at the bottom of the slice, you can see the roots coming out. And so that's an indication that we've got roots at all, which is important. Roots are going to do a lot to hold the soil in place, and they also support a lot of the microbial biology. Roots exude a lot of sugars, which is a source of food for bacteria in the soil, and also for fungi as well. And they can support a lot of beneficial partnerships that kind of keep the nutrient cycling in soil going and all of the good biological stuff. And then the direction is the other thing that we want to think about. So if you
So water infiltration rings can be really helpful. The other thing to note is that when you're hammering those into the ground, you usually use a board on top. You want to try and get them straight in. And you're also considering that smoothness I was just talking about that we make with the shovel. When we hammer the rings in the ground, we could be influencing the way that water moves along that metal ring. So sometimes tucking in the top of the ring, there are little tricks that you can do, and you want to be really consistent with it. And this is another reason why 3 to 5 is important, because it's not likely that you're going to make the same mistake on all 5.
You got a camera, you know.
All right, so I'm going to say I am the landowner, and do you want to be our assessor? So you're the extension person who's coming out to my farm, and I want to ask you, how can I improve water infiltration on this hillside so that I don't get runoff down the road, but I still get the water, but not the silt and road in my stream? What might you say? There's no wrong answer. I just made that whole question up.
I would, well, geez, we've already got pretty good cover, so, you know, I think high points for surface cover that would at least slow the movement of water and promote infiltration. What else can we do? So I don't think we need to change the vegetation.
Water the living roots, the roots on top.
Thank you.
And if someone wants to identify a place where there is a leguminous plant,
Likely going to be clover. That's most noticeable time of year.
So we got a bit here.
A little bit there. And we can dig that up. You all still have the lenses. Yeah, hang on to those because we're going to look at clover and see if we can find some nitrogen nodules. great. And if we don't find them, I did go out to the oat and pea patch earlier and got some nice peas that are up on the table in there with nitrogen nodules.
We also have about 10 minutes and we've been given permission to walk around to a different spot if we want to do that too.
Okay.
Let me add something here. One thing that happens is sometimes people get really excited. They find the legume, they tug the legume out. That'll often just strip all the nodules off,
Right
so you'll
off
never
of it.
see them.
That's
So you have to
true.
really tease the soil apart.
That's right, because they're little balls.
They're little tiny things.
Yep.
We all think they're going to be the size of peas, but they're not. They're tiny.
I once saw nitrogen nodules the size of a marble.
No kidding.
And it was on a multi-species cover crop on a small organic farm in central Vermont. And they were growing just for the soil. Okay, you can start to see them. Do you see it?
Yeah.
Squint.
Yeah.
Squint.
Are they black or what color? are they?
Like this little thing?
Yeah. Yeah. Here, take that in a hand lens and have a look and pass that around. Those can get quite large. And my understanding of the nitrogen cycle is a little bit limited. But the way I understand this works is that in order to fix nitrogen in the soil, first you have to have a leguminous plant, and they partner with rhizomaceous bacteria or rhizobium bacteria, which is rhizosphere, so think root system. These are little microbes, bacteria that live in the root system. They're able to magically pull nitrogen out of the air and sequester it in their bodies. And they create these little anaerobic situations inside these cellular, like that's the little ball you're looking at. I'm not a scientist.
Nodules.
Can you tell?
Yeah.
Thank
nodules.
you. Nodules. And in order for it to fix the nitrogen in a usable form, so turning it from nitrate to nitrite, the 2 to the 3, which one is usable by perennials, one is used by annuals. So in order to get that full cycle, you've got to have 2 of these rhizobium species in order to fix the nitrogen. If your nodule has pink on the inside, which is really hard to see on ones these small, the pink is an indicator that the nitrogen has been fixed and is now usable. You got one, Peter?
So,
And it's pretty cool to see the pink stain inside. And the more you look for them, you will find them. And yeah, you can. Sometimes, like if you hold it up to the light, and depending on
I can't tell.
a bunch of different things.
Seems
right
like
here.
it is.
And I am always surprised by places where I'll often tell people to go. We'll go out and collect samples, and I'll say,
there's
well, I
one.
want you to collect a
There's one
sample
right
from
there.
where you
Yep.
see the soil health principles in action, and a place where you
There's a few.
don't.
There's 1, 2,
Yeah,
3, 4,
yeah, yeah.
5.
Good.
Plants, but any plant that has a taproot has the ability to break through hardpan at some level. So dandelions are actually really helpful in our lawns. They're great aerators.
I would want them to at least start trying to figure out how to cover crop. Because I think that's a good entry point. You're not, in this particular case, saying don't till anymore is like not going to sell. But just trying to figure out how to get a cover crop in there.
You have one bed of cover crop over there.
Yeah.
So it's
but
interesting why they're not here. You know?
if they were tarping it, maybe they're, they have plans for it. So, but it's just a little spot. Wouldn't be hard to even, like when you're here visiting and you have your pocket full of seeds.
There's like a rolling. Hi friends. Sorry, can you start moving back in after your last thought, Heather?
Well, I lost it now. I'm kidding. Well, I remember now, but I think the more farms you visit, the more you see, right? And I just told you about what we do at our farm, and it works for us. And so now when you talk to Farmer Jake, you can say, you know, I was at this other farm, very similar, and you know what? One of the things I saw that they were doing was this. Is that something, have you tried that? You know, just gaining that information